Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss how insulin works. In the previous video, we discussed how insulin affects the human body, focusing mostly on the liver, the skeletal muscle, as well as the white adipose tissue. We saw that insulin increases glucose uptake by cells from the bloodstream. Insulin promotes synthesis. So, for example, insulin increases glycogen synthesis. Insulin increases triglyceride synthesis. Insulin increases protein synthesis. However, insulin decreases glucose synthesis or gluconeogenesis. And that makes sense because insulin is secreted after a meal. There is high blood glucose, and so we do not need the liver to make more glucose. While promoting synthesis, insulin will decrease breakdown. So insulin, for example, will decrease glycogen breakdown as well as triglyceride breakdown. So that's basically what we discussed last week, the effects of insulin on the human body. How does insulin carry out all of these effects? Let's see how insulin carries out all of these effects. Insulin secreted by the beta cells of the pancreas will travel in the bloodstream to every part of the body. And if there is receptors on the cell present for insulin, what we call the insulin receptor or IR, then the insulin signal will bind the receptor. If there's no receptors present, the insulin basically will simply go back into the bloodstream. Once insulin binds the receptor, this triggers a signal transduction pathway, an insulin transduction pathway. So here insulin is the signal. This signal transduction pathway can then go and change the catalytic activity of existing enzymes or functions of various proteins, as well as change the amount of enzymes or proteins in the cell. So, for example, we'll see how insulin can cause the sequestering of enzymes as well as transport proteins in certain compartments. For example, we'll see how the GLUT4 transport protein travels from the cytoplasm to the plasma membrane in case of muscles and adipocytes. We'll see examples of glucokinase, the enzyme in liver, being transported between the nucleus and cytoplasm. We'll see how insulin carries out allosteric regulation. We'll see how insulin carries out covalent modification, especially phosphorylation and dephosphorylation of key metabolic enzymes. And we'll see examples of how insulin can change the concentration of existing enzymes and proteins, especially by increasing or decreasing transcription of that protein. Let's first see how insulin stimulates glucose uptake by cells in the liver, muscle, as well as white adipocytes. First, the liver. Due to the anatomy, the liver will see a much higher insulin concentration, almost a threefold higher of insulin concentration than other organs in the body. The major glucose transport protein of the liver is GLUT2. Insulin doesn't have any significant effect on this transport protein, on GLUT2. And we'll see this is very different than the effect of insulin in muscles as well as adipose tissue. We'll see that insulin has a very dramatic effect on GLUT4. But this is not the case in liver, which has GLUT2. However, when GLUT2 transports the glucose from the bloodstream into the cell, into the hepatocyte, this glucose is immediately converted into glycogen. So this means the concentration of glucose within the cell is kept very low. And since the concentration of glucose outside the cell in the bloodstream, remember after a meal we have a lot of glucose that is being absorbed. So this glucose concentration is high, glucose concentration in the cell is low, the concentration gradient is very large, and that will cause more and more glucose to enter the hepatocyte. So increased glucose uptake in the liver is mostly due to concentration gradient difference. Let's now look at glucose uptake in muscles and adipose tissue. In case of muscles and adipose tissue, the major glucose transport protein is GLUT4. And these proteins are found in vesicles inside the cytoplasm. So what happens after a meal, when insulin is released, the insulin will bind the insulin receptor that will trigger a signal transduction pathway. And one of the effects of this pathway is this enzyme, protein kinase B, also called AKT, will become activated. This protein PKB will then go on to modify, phosphorylate several proteins that are involved in vesicle trafficking, basically sending the vesicles from the cytoplasm to the plasma membrane. And this causes an increased movement of GLUT4 from the cytoplasm to the plasma membrane. So GLUT4 will now get incorporated or embedded in the plasma membrane, and now the glucose in the bloodstream can start entering the muscle cell, myocyte, or the adipocyte. So this is an example of insulin affecting the sequestration of a protein within a certain compartment. Once the glucose is in the cytoplasm, then it can be converted into glycogen or protein in case of muscle, or it can be converted into fatty acids in case of the adipose tissue. And so this will make the glucose concentration in the cytoplasm of these cells very low compared to the concentration in the bloodstream, which is very high after a meal. And this difference in concentration will lead to a concentration gradient which will encourage glucose to enter the cell. Now let's look at the effect of insulin on glycogen synthesis. How does insulin increase glycogen synthesis, which is also called glycogenesis? And this happens mostly where most of your glycogen is stored, and that is your liver as well as your muscles. A key enzyme for glycogen synthesis is glycogen synthase, or GS. As you can see, the GS can exist in the relaxed form, which is the very active form, or it can exist in the T or tense form, which is the inactive form. Glucose 6-phosphate is a very powerful allosteric activator of glycogen synthase. So it will convert the T form into the R form. So whenever there is a lot of glucose 6-phosphate, the inactive glycogen synthase becomes very active glycogen synthase, and that will lead to increased glycogen synthesis. Now, insulin increases the transcription of glucokinase, GK, also called hexokinase 4, that is found in the liver, and it also facilitates the transport of glucokinase from the nucleus to the cytoplasm. So not only do we have more glucokinase, but this glucokinase is transported from the nucleus to the cytoplasm. Once it's in the cytoplasm, the glucokinase will convert glucose to glucose 6-phosphate. Concentration of glucose 6-phosphate increases in the hepatocyte, and this glucose 6-phosphate will then go and allosterically activate glycogen synthase, and that increased activity of glycogen synthase will then lead to increased glycogen synthesis or glycogenesis. So the rapid conversion of glucose into glycogen is once again going to keep the concentration of glucose very low in the hepatocyte compared to the concentration of glucose in the bloodstream, which is very high after a meal. Once again, that will lead to a concentration gradient, and that concentration gradient will lead to more glucose from the bloodstream entering the cell, and that glucose being converted to glycogen. So there is increased glycogen synthesis flux due to this dependence of glucose transport together with glycogen synthesis. A similar effect happens in skeletal muscles. Insulin increases the transcription of hexokinase 2. This is the major form of hexokinase found in skeletal muscles, and so the glucose will be rapidly converted to glucose 6-phosphate. This concentration will rise. Glucose 6-phosphate will then activate glycogen synthase, and that will lead to increased glycogen synthesis. Besides allosteric activation of glycogen synthase, insulin also increases glycogen synthesis by increasing the dephosphorylated, which is the active form of glycogen synthase. We'll represent that as GSOH. Let's see how this happens. Now, glycogen synthase can exist in the non-phosphorylated or the dephosphorylated form. You can see the OH group here, or it can exist as the phosphorylated form, and we see the hydroxyl groups becoming phosphorylated, and these hydroxyl groups are present mostly on serine residues of glycogen synthase. Now note that the dephosphorylated or non-phosphorylated form is the active form. We'll just call that the R state, while the phosphorylated form is the inactive or the T state. The phosphorylation of glycogen synthase, which will convert the active R form into the T form, inactive T form, that is carried out by several proteins. We'll just talk about two important ones, GSK3 as well as pKa, while the dephosphorylation, the conversion of the inactive T form to the active R form, that is carried out by a protein called PP1. So insulin can increase the active dephosphorylated glycogen synthase either by decreasing phosphorylation, decreasing this direction of the reaction, the phosphorylation part, and that can be done by inhibiting GSK3 or inhibiting pKa. Or another way insulin can increase the active dephosphorylated glycogen synthase is by increasing the reaction in this direction, basically increasing dephosphorylation. And since dephosphorylation is carried out by PP1, this can be done by activating PP1. Let's first see how insulin decreases glycogen phosphorylation by inhibiting glycogen synthase 3 or GSK3. GSK3 itself exists as the non-phosphorylated active form or the phosphorylated inactive form. When insulin binds its receptor, that leads to a signal transduction that will activate protein kinase B, PKB, and protein kinase B will convert the active GSK into the inactive phosphorylated GSK3. And that will lead to a reduction in phosphorylation, which means there will be more active glycogen synthase that will lead to higher glycogen synthesis. Now let's see how insulin reduces the activity of pKa. Now protein kinase A is very strongly activated, allosterically activated by this chemical called cyclic AMP. Now cyclic AMP is broken down into AMP by an enzyme called phosphodiesterase 3B or PDE3B. When insulin binds the receptor, that will trigger the activation of PKB, and this protein kinase B will basically activate this phosphodiesterase, thereby breaking down cyclic AMP into AMP. So without cyclic AMP, there will not be allosteric activation of pKa, this enzyme will not work, phosphorylation will slow down, and that will lead to an increase in active glycogen synthase, leading to increased glycogen synthesis. So besides slowing down this phosphorylation step, another way to increase active glycogen synthase is to increase the dephosphorylation step. So let's see how this happens. Now remember that glycogen metabolism, whether it's the synthesis or the breakdown of glycogen, is happening on the glycogen particle found in the cytoplasm. So how does insulin increase the dephosphorylation? What insulin does is when it binds to the receptor, that will lead to the transfer of this protein PP1 from the cytoplasm to the glycogen particle. So when PP1 is targeted to the glycogen particle, now it can start dephosphorylating, remove these phosphates, converting inactive glycogen synthase into active glycogen synthase. So the concentration of active glycogen synthase will rise, and that will lead to increased glycogen synthesis. Now let's look at the effect of insulin on glycogenolysis. So while insulin increases glycogen synthesis, insulin will also at the same time decrease glycogen breakdown or glycogenolysis. Let's see how that happens. Now the key enzyme for glycogen breakdown or glycogenolysis is this enzyme glycogen phosphorylase, which we shall abbreviate as PYGL. Once again, PYGL exists in the active R form or the inactive T form. In case of the liver, as glucose is taken up into the cells by GLUT2, the concentration of glucose in the hepatocytes will increase. Now glucose is an inhibitor. Glucose allosterically inhibits this glycogen phosphorylase converting the active R form into the T form. And this way, it will reduce the activity of glycogen phosphorylase, and thereby it will reduce glycogen breakdown. So it will reduce glycogenolysis. In case of the muscle cells, the inhibitor of glycogen phosphorylase is glucose-6-phosphate. Glucose-6-phosphate is a very powerful inhibitor of glycogen phosphorylase. Remember, glucose-6-phosphate activates glycogen synthase. However, it's an inhibitor for glycogen phosphorylase. So as G6P concentration increases in the muscle, glycogen synthase will be activated, but glycogen phosphorylase will be inactivated. So the R form will be converted to T form, thereby reducing glycogen breakdown, while at the same time causing glycogen synthesis to increase. Besides allosteric inhibition, insulin can also cause covalent modification, thereby increasing the inactive dephosphorated or non-phosphorated form of glycogen phosphorylase. Let's see how that happens. Now, glycogen phosphorylase, PYGL, once again exists as the dephosphorylated form or the phosphorylated form. However, unlike glycogen synthase, the active form is the phosphorylated form, while the inactive form is the non-phosphorylated form. So insulin increases the inactive form, the inactive form, either by decreasing phosphorylation, so by decreasing phosphorylation, or increasing dephosphorylation. A major protein that carries out phosphorylation is this enzyme PHK, or phosphorylase kinase. So insulin can inhibit, or insulin inhibits PHK, thereby inhibiting phosphorylation. The protein that causes dephosphorylation, like before, is PP1, phosphoprotein phosphatase. So insulin can increase dephosphorylation, and by increasing dephosphorylation, the active form is converted into the inactive form, thereby decreasing glycogen breakdown. So let's see how this happens. Insulin binding the receptor results in the targeting of the glycogen particle once again by PP1, phosphoprotein phosphatase 1. And one of the effects of PP1 is to basically convert the phosphorylated form of PHK, phosphorylase kinase, which is the active form, into the inactive form. So basically it's inactivating this enzyme here, thereby stopping phosphorylation. And that will increase the concentration of inactive PYGL, which will result in the decreased glycogen breakdown. So besides inactivating PHK and thereby reducing phosphorylation, PP1 being targeted to the glycogen particle also causes another effect, and that is the PP1 at the glycogen particle will dephosphorylate this active PYGL into inactive PYGL. And now this inactive glycogen phosphorylase will lead to the decrease of glycogen breakdown. So that is how insulin can decrease glycogen breakdown. Now let's look at how insulin decreases glucose synthesis or gluconeogenesis, which is predominantly in the liver. Now, there are two very important transcription complexes which increase the transcription of key gluconeogenesis enzymes. So the faster the transcription of the gluconeogenesis enzymes, the higher the concentration of gluconeogenesis enzymes, and the higher the concentration of gluconeogenesis enzymes, the faster gluconeogenesis will happen. One of this complex is a short-term fasting complex, and this involves the protein CRAB, which is camp response element binding protein. It also involves the protein CRAB binding protein, or CBP, as well as a co-activator required for this transcription. This is your CRAB-regulated transcription co-activator 2, or CRTC2. Another transcription complex is the long-term fasting complex, which involves the protein Fockhead box protein 01, or FOXO1, as well as its co-activator, peroxisome proliferative activated receptor gamma 1 alpha, or PGC1 alpha. So these two transcription complexes will increase the copying of the DNA in the nucleus into mRNA, and that increase in mRNA will lead to increase in gluconeogenesis enzymes, and there will be increased gluconeogenesis. So how insulin decreases gluconeogenesis is by decreasing the synthesis of the key gluconeogenesis enzymes, especially glucose 6-phosphatase, as well as PEP carboxykinase. So by decreasing the concentration of these enzymes, gluconeogenesis slows down or even stops. So let's see how this happens. So basically, insulin will stop these complexes from working. So for example, insulin activates a protein called salt-inducible kinase 2, or SIK2. This protein then goes on to phosphorylate CRTC2. CRTC2 that is phosphorated cannot enter the nucleus from the cytoplasm, which means the gene for gluconeogenesis is not transcribed into mRNA, which means gluconeogenesis enzyme is not available, and thereby stopping gluconeogenesis. Insulin also activates protein kinase B, which then goes on to phosphorylase FOXO1. Now FOXO1 now cannot enter the nucleus, and once again the transcription will not happen, the genes will not be transcribed into mRNA, gluconeogenesis enzymes will not be formed, and there will be no gluconeogenesis. So here's a good example of how insulin prevents transcription and thereby prevents the formation of an enzyme or of enzymes. In case of gluconeogenesis, these two enzymes, and without these enzymes, gluconeogenesis will not happen, and that's why we do not see glucose synthesis in the liver after a meal when insulin levels are high. So far, we have seen how insulin increases glucose uptake by various cells. We saw how insulin increases glycogen synthesis, especially in the liver and the skeletal muscles. We saw how insulin decreases glycogen breakdown, once again, mostly in the liver and your muscle. And we saw how insulin decreases gluconeogenesis, especially in the liver. In the next video, we shall look at the effects of insulin on the metabolism of triglycerides as well as proteins. So until the next video, once again, thank you very much for watching. Have a wonderful day, and we will see you in the next video.